In the next 5 minutes, I'll show you everything you need to know to load a SPRQ-Nx SMRT Cell and start a run. And if you're already a Revio user, you'll see that not much has changed from what you're used to. Any SMRTbell library can be run with Spark chemistry or SPRQ-Nx chemistry. The library prep remains unchanged. SPRQ-Nx takes advantage of the on-market Spark polymerase kit while bringing in new SPRQ-Nx sequencing plates and SPRQ-Nx SMRT cells. You might choose SPRQ-Nx if you're looking to sequence many samples or if your research demands very high coverage. It's pretty easy to tell the kits apart. All SPRQ-Nx consumables come in gray boxes, whereas Spark consumables are magenta. So let's head to the lab and I can show you how easy it is to set up and load a run. Our polymerase-bound SMRTbell libraries are ready to go. So now we'll move on to preparing the sequencing consumables. We start by thawing our SPRQ-Nx sequencing plates in a room temperature water bath, protected from light, for 1 hour. While those thaw, make sure you hang on to the QR code from the sequencing plate box. We'll use it in the next step to link the consumables with our run design. Run design is pretty much the same. The only change is that we'll now select SPRQ-Nx for the sequencing plate. Today, we're going to set up day 1 of a 24 acquisition run with SPRQ-Nx. That means we'll need 2 SPRQ-Nx sequencing plates, each containing 4 libraries. Then we're going to load 1 SMRT Cell Next tray, which contains 4 multi-use SMRT cells. If the computer where you're setting up the run design has a camera, it's easiest to scan the included sequencing plate QR code. This links the sequencing plate with the run design. Once everything looks good, be sure you hit save before moving on. Once the sequencing plate is thawed, loading your samples is the same. We start by piercing the foil for A1 through D1 sample wells, and then we add our polymerase-bound libraries into the respective wells. To prevent evaporation, apply the foil seal strip over the sample column. And with that, we're ready to load the Revio. To start our SPRQ-Nx sequencing run, we place our prepared SPRQ-Nx sequencing plates on the work deck. The system will scan our sequencing plates and automatically pull up our linked run design. Then, we'll load 1 SMRT Cell Next tray onto the work deck. With standard Spark chemistry, you get 1 acquisition per SMRT cell. But with SPRQ-Nx, we can use the same SMRT cell for multiple acquisitions. Let's double check that everything looks correct with the samples and the run setup, and then we can start the run by clicking Continue and closing the door as usual. As the Revio system proceeds through our sequencing run, it will start by preparing and sequencing the first set of samples on each next SMRT cell. After those acquisitions are complete, the instrument automatically washes the next SMRT cells and prepares them for the next set of libraries. There's no user intervention. It's all handled on the instrument. With SPRQ-Nx, we retain the ability to preload the Revio system. This way, when our run finishes, the next preloaded consumables will begin sequencing immediately, even if it's 2 in the morning. And with that, we're done. It really is that easy. If you're a seasoned Revio user, you'll notice that SPRQ-Nx is very similar to Spark. Not much has changed. And if you're new to Revio, you can see how simple it is to set up a SPRQ-Nx run.